Photosynthesis rates vary under different colored lights and are the slowest under green light, by Olivia Postel and Yasmin Sharbaf. Background information: photosynthesis. Photosynthesis is required for most life on Earth, is catalyzed by sunlight. It may not be required directly, as it is for many plants and organisms that perform photosynthesis, but it's also the source of gaseous oxygen, and it helps get carbon dioxide out of the atmosphere so that organisms like humans can breathe. So, photosynthesis is performed by different pigments that absorb wavelengths, and some examples of the pigments are chlorophyll A and B. And the basic steps of photosynthesis are: 1, absorption of the light by plants. So the light, the photons, will hit the plant and they'll go into photosystem 2. And then electron transport and reduction of NADP plus to NADPH. Then generation of ATP from that. And then the last step is carbon fixation, which is carbon dioxide being converted into carbohydrates. This process is incredibly important, not only for growing food and such, but also about global warming and how some of this carbon dioxide that has been fixed from the atmosphere and stored in things such as fossil fuels is now being released at a rate much greater than it can be recovered by photosynthesis. So, background information about thylakoids. Thylakoids are the center for light-dependent reactions. Mitochondria may be the well-known powerhouse of the cell. You always hear that in biology, and yet for organisms that perform photosynthesis, the thylakoid is basically the mitochondria. It's the powerhouse of the cell. So, unlike mitochondria, chloroplasts contain a third membrane, which is the thylakoid membrane, which we were specifically looking at. And that's the site of photosynthesis. And on this membrane, there are proteins and pigments that allow for the absorption of light. And the rate of photosynthesis can be determined using the Hill reaction, which is demonstrated on the bottom of this slide. We used DCPIP to help with this, which is blue when oxidized and colorless when it has been reduced.
So now some background information about light in plants. So there are many factors that can affect photosynthesis, including temperature, how much CO2 is available, water, and also how much light is shined into the plant, or how much light the plant or the chloroplast is absorbing. And when in light, there is a visible spectrum that the human eye can see, and that is a range from around 400 to 700 nanometer. And for example, when we look at the green spinach leaves that we use in our experiment, they are green because they absorb long and short wavelength, which correspond to red and blue colors, while it reflects, and I mean the chlorophyll reflects medium, which is green wavelength. And also some experiments that relate light and colors to plants is that they found that putting plants under a red colored net boosted their growth rate.
Our experimental question was: how does the rate of photosynthesis in spinach thylakoids change as the light intensity is kept the same, but different colored filters are placed over the light? Our independent variable is the color of light reaching the thylakoids, and that's determined by the filter that we placed over the light source. We tested red, blue, green, and purple, and then we also did a test without any filter at all. Our experimental design. First, we prepped the thylakoid stock solution from spinach. Then, we added 50 microliters of thylakoid suspension to 5 milliliters of clear resuspension solution in a 13 millimeter test tube, covered it with parafilm, inverted it once, and blanked the spectrophotometer using that. We made sure to set the spectrophotometer at 580 nanometers, and we blanked that first to a normal light without using any sort of colored filter, and plotted it in our data points in a graph. I'll describe how later, just to make sure that the reaction was consistent and it wasn't going too fast. And so we determined that the light intensity should be set at 64 micromole, nanometers per micromole. And so every time we used a different filter, we had to blank the instrument once more using the blank that we had prepared. And moving on to when we tested the colors. First, we put whatever colored filter we were testing over the light. We blanked it, and then right before we tested the tube, we added 50 microliters of thylakoid mixture to 5 milliliters of the reaction mix that contained the DCPIP, exposed it to colored light for 10 seconds, and then put it inside the spectrophotometer for 5 seconds for reading. And we exposed it again for 10 seconds, and then read for 5 seconds, and we repeated that 7 times until we had 7 data points for each test tube, and we recorded the measurements. We had to do this several times for each of the colors. Reasons we'll explain later. We were having a little bit of trouble with with getting consistent numbers.
Results. The figure shows that the rates of photosynthesis differ under different colored lights, which are green, blue, purple, red, and no filter or the natural light condition. All rates were measured under 64 nanometer over micromole light intensity and over a period of 90 seconds. Our graph shows that the green light had the shallowest regression line of them all, and as we can see, the dots are very scattered throughout the graph, which is something we expected. On the other hand, purple had the fastest reaction rate or the absorption rate of all the other of all the colors. Although it's not that much different than the red light, it is a bit faster than it. I also included the equation and R on the right of the graph because it looked a bit cluttered.
Results continued. Figure 2, the average rate of photosynthesis for each color filter was slowest for green and fastest for purple. So this graph shows the coefficient or the slope of the best fit line for each graph that we obtained for each colored sample. As we expected, when the green filter was put over the light source, the reaction occurred the slowest. However, we had expected the red filter to cause the reaction to go the fastest, and that was not the case. The reaction actually occurred the fastest when the purple filter was placed over the light, and it occurred much, much faster than any of the other ones. All of these were measured under 64 nanometers per micromole light intensity, and the coefficient slope is indicated on the graph.
Discussion. So in the experiment, we encountered many that resulted in variabilities and inconsistency in our results, such as measuring the testing tube multiple times because it had some drops of water in it, and so we will take it out, wipe it, and then put it back, and then it will give us a completely different measurement. Or even when we open the lid and then close it, it will give us a different reading. We also tested the colors multiple times and got completely different measurements most of the time. We did that because the graph sometimes looked weird and we were not sure whether the reading was correct or not. We also, when we encountered unexpected outliers, we would repeat the measurement over, and of course we would prepare a different testing tube, and so that resulted in each color being tested up to 3 times. Also, the light intensity. So we use 64 micromole light intensity, but sometimes when we put the device that read the light intensity, it will read 64. But when we try to read it again to make sure that it is 64, it will give us a different measurement, and so we had to readjust it. So we were not sure whether that was the correct light intensity or not. Conclusion. So although we did not achieve perfect results and we ran into many problems with the spectrophotometer, we noticed in our results that the green filter had the shallowest slope, which means it had the slowest reaction rate and absorption of all the colors. On the other hand, the purple light had the steepest slope and the fastest reaction, or at least until the DCBIP dye ran out and the reaction speed plateaued. And that was a bit different than our hypothesis. So our hypothesis initially was that we'll see the fastest reaction in red, which we did not see in our results because we saw that purple had the fastest reaction. But we did see that the green filter had the slowest reaction, as we see in the results. Logical explanation. So our reasoning behind our results is that first, since spinach leaves are green because they have green chlorophyll, they reflected the green color while they absorbed other colors, and that's why we saw the slowest absorption rate in green. And also a study showed that a photosynthesis organism had a peak absorption at 625 nanometer, which is the red wavelength, because electron transport rate at the photosystem 2 increases with wavelength. A second study showed that red and blue lights can improve photosynthesis. And our results does show that purple, which is associated with the blue light, had the highest absorption rate, while green had the least absorption. However, we did not see that in the red. Also, light efficacy is important to increase photosynthesis rate because it regulates electron transportation. But since our light intensity was very inconsistent, it might have been low at some points or high, our results might be inaccurate. Future directions. So first and foremost, we want to eliminate any variabilities or inconsistencies in our future experiments. Things we are interested in testing is how different light intensities affect the rate of photosynthesis over different colored filters. So instead of using only the 64 micromole light intensity, we can experiment with lower or higher light intensities and see how they affect absorption rate over the different period of times. We also can test non-green plants instead of using the spinach leaves, which are green, and see how their absorption rate is affected by the colored filters. And so we think this experiment was important because we might be able to apply these results or our future results to use them in greenhouse windows. For example, we might be able to use specific colors in the windows in order to increase the photosynthesis and the growth rate of the crops we eat. And so that's the end, and thank you so much for listening.